Hi, we're continuing with my notes on life science and cell biology in a nutshell. So we're learning about the various important organelles within cells. We just learned about lysosomes. So here we have staining showing what lysosomes look like. The catabolic hub of the cell is the lysosome. So they break down macromolecules, they degrade materials. Pathological consequences may result as a result of undegraded accumulation of materials that may be toxic, etc., toxic proteins, etc., that are not broken down like they would need to be. So here again, showing some of the the organelles. So we have the nucleolus containing a ribosomal subunit, content. So that's where the ribosomal subunits are made. And then you have ribosomes, free ribosomes, and bound ribosomes are on the endoplasmic reticulum, which come out from the outer membrane of the nucleus. So the rough ER contains ribosomes bound to its membrane sacs. Smooth endoplasmic reticulum doesn't have ribosomes. Instead, it's smooth. It is only in charge of lipid and steroid production, while the ribosomal units on the ER are what produce proteins and enzymes, etc. So the cell also has a cytoskeleton or microtubule network, which help with its shape. So Golgi apparatus, which we learned about previously, are associated with the endoplasmic reticulum in that they receive proteins and lipids made by the ER to mark them, label them by modification, adding of phosphate, sugar, and amino groups, etc. Whatever needs to be done to appropriately mark the molecules to be destined to specific locations, either inside or outside the cell. So the molecules, proteins, or lipids may be headed to various organelles or to the membrane of the cell to be secreted via vesicles to the outside of the cell environment. And then lysosomes are a high pH environment sacs which break down molecules, macromolecules, even organelles, parts of the cell, or even bacteria or anything that's taken up by the cell that needs to be broken down and is not wanted or needed. So the lysosomes are the digestive system of the cell, containing enzymes breaking down proteins or even complete microorganisms. Centrosome and centrioles, part of the cytoskeleton as well, help with cell division and cleaving of cells, one cell into two, etc. Here, a color-enhanced transmission electron micrograph showing the lysosome at 30,000 magnification. Lysosomes are cellular organelles containing acidic hydrolase enzymes, breaking down waste materials and cellular debris. The name lysosome derives from the Greek words lysis, to separate, and soma, body. Lysosomes prevent accumulation of malfunctioning proteins by continuously breaking them down into their constituent amino acids, which are in turn used to build new proteins. So they break down all kinds of micromolecules into their subunits to be recycled. The red dots here are also lysosomes. The green is a neuron that has died. So the lysosomes help with the recycling. The lysosome contains multiple proteases or enzymes, and it accounts for 20% of protein turnover in cells. Lysosomes degrade organelles and proteins of the membrane and various proteins. Cytoplasmic proteins can also be degraded through autophagy, a process in which organelles and bulk cytoplasm are enveloped in double membranes delivered to the lysosome subsequently. So, lysosomes are a heterogeneous population of membrane limited vesicles or bubbles, differing in size and density, are identified and characterized first in the mid-1950s by De Duve scientists. There are now several fluorescent probes used to visualize and assess membrane traffic to the lysosome, as well as probes assessing the activity of lysosomal hydrolases in cells that are alive. More novel proteins and key cellular functions are associated with lysosomes. Lysosomes are small vesicles derived from the Golgi apparatus containing up to 50 acidic enzymes or hydrolases at a pH of 5. These enzymes include lipases, proteases, nucleases, sulfatases, etc. So depending on what the enzyme breaks down, it can be lipids, proteins, nucleic acids such as DNA and RNA, etc. Primary lysosomes are inactive organelles typically dispersed throughout the cell. The lysosome fuses with the phagosome. A secondary lysosome is formed, then is activated and begins the digestive process. After completion of the digestive process, components that cannot be broken down by lysosomal enzymes form membrane-bound inclusions termed residual bodies, which are secreted as in macrophages or retained within the cell as in nerve cells. Over time, the accumulation is visible in stained nervous cell tissue and termed lipofuscin granules or aging pigment. So co-stained with lysosome dye, lysobright red, showing and phalloidin in green, to label actin filaments, nucleic stain in blue, nuclei, so the nucleus. The cells are fixed in 4% formaldehyde. This kit is used for studying, including cell adhesion, chemotaxis, multi-drug resistance, cell viability, apoptosis, and cytotoxicity. So it's very important to know what the lysosomes do and how materials are broken down within the cell, especially toxic proteins or molecules that may harm the cell. over time. Thank you.